Welcome to this fascinating journey into embryonic brain development. Today, we are going to explore how you, and specifically your brain, began. It is hard to believe, but the most complex organ in the known universe starts out as nothing more than a simple, flat sheet of cells. We will uncover the biological magic and the elegant mathematics that drive this incredible transformation from a tiny tube into a fully functioning human brain. To understand how the brain forms, we must first look at a process called neurulation. Why does this matter? Because before you had a face or a heart, your entire nervous system was just a flat sheet of dough-like cells on the embryo's surface. During the third week of development, a specific patch called the ectoderm thickens into a neural plate. Imagine pushing down the center of this flat sheet to create a groove, while the edges naturally fold upward. This physical folding is the absolute foundation of all neuroscience, setting the stage for the three-dimensional structure of your brain and spinal cord. Now that the edges of our flat sheet are folding upward, what happens next? Those rising edges, the neural folds, eventually meet in the middle and fuse together. This creates a closed, hollow cylinder known as the neural tube. The cells inside this tube will multiply to become your entire central nervous system. Interestingly, the hollow space inside does not just disappear. It eventually forms the fluid-filled ventricles in your brain and the central canal of your spinal cord. This physical layout is the blueprint for everything that follows. Building on the formation of the neural tube, we must understand what happens when this process goes wrong. Complete and perfect closure of this tube is critically important for survival. If the neural tube fails to close completely during embryonic development, it results in severe and often fatal birth defects known as neural tube defects. For example, if the top part of the tube fails to close, the brain cannot develop properly, resulting in a condition called anencephaly. On the other hand, if the bottom part fails to close, the spinal cord is left exposed, resulting in spina bifida. This highlights just how delicate and crucial this early folding process truly is. With our closed neural tube fully formed, how does it transform into a complex brain? Imagine blowing air into a long, skinny balloon, but pinching it near the top. The front end, or rostral end of the neural tube, does exactly this, swelling into three distinct bubbles called primary brain vesicles. The first swelling is the forebrain, which will become your cerebral cortex, the seat of conscious thought. The middle swelling is the midbrain, a crucial relay station for signals. The third is the hindbrain, responsible for vital functions like breathing. As development continues, these regions subdivide further into 5 secondary vesicles, mapping directly to the adult brain. Creating the physical rooms of the brain is only half the battle. We now need to fill those rooms with inhabitants. The human brain requires a staggering 86 billion neurons to function properly. All of these must be generated from a very small starting population of stem cells located inside the neural tube. This rapid creation of new neurons is known as neurogenesis. To achieve such massive numbers in a very short embryonic timeframe, these cells cannot just add up one by one. Instead, they must multiply exponentially, doubling their population over and over again. To understand how this explosive cellular growth happens, let us look at the mathematics behind neurogenesis. This formula models how a small group of cells grows exponentially over time. N of t represents the total number of cells at a given time. N0 is the initial number of stem cells we start with. The variable t is the total time that has passed, and d represents the doubling time, which is how long it takes for one cell to divide into two. Physically, if the total time increases, the exponent grows, causing the cell population to skyrocket. Conversely, if the doubling time increases, meaning cells take longer to divide, the overall rate of growth slows down significantly. Now that we have our exponential growth formula, let us see this with a concrete example. We are given a tiny section of the neural tube starting with exactly 100 stem cells. These cells have a rapid doubling time of 12 hours, and we want to find the total population after 72 hours. First, we figure out how many complete doubling cycles occur by dividing the total time, 72, by the doubling time, 12. This gives us 6 complete cycles. Next, we substitute our initial population of 100 and our 6 cycles into the formula. 2 to the power of 6 is 64. Multiplying 100 by 64 gives us a final answer of 6,400 cells. Physically, this means in just 3 days, a tiny cluster explodes into thousands of cells, which is necessary to build the brain. So, we have successfully generated thousands of cells, but this brings us to an important question. How does a specific cell know whether to become a motor neuron or a sensory neuron? The answer lies in chemical signals called morphogens. Imagine standing near a campfire. The closer you are, the hotter it feels, and as you walk away, the heat fades. Cells in the neural tube use a similar distance trick. They measure the concentration of specific chemicals secreted by neighboring tissues. The concentration of these morphogens tells the cell exactly where it is located in the developing brain, which in turn dictates its final identity and function. To understand exactly how these chemical signals fade, we use a mathematical model for continuous decay. This formula describes how a morphogen's concentration drops over a distance. C of x represents the concentration at a specific distance x from the source. C0 is the maximum concentration, right at the source, where the chemical is produced. The constant E is the base of the natural logarithm, used to model continuous decay. Finally, lambda represents the decay length, which is how far the chemical can effectively travel. Physically, if the distance x increases, the concentration drops exponentially. If the decay length lambda is large, the chemical travels further, influencing cells much deeper in the brain tissue. Now that we have our decay formula, let us apply it to a famous morphogen called sonic hedgehog. We are given a source concentration of 1,000 molecules, a decay length of 50 micrometers, and a cell located 100 micrometers away. We need to know if the concentration reaching this cell is above the threshold of 100 molecules required to become a motor neuron. We substitute our values into the formula. The exponent becomes negative 100 divided by 50, which simplifies to negative 2. The constant e raised to the power of negative 2 gives a decay factor of roughly 0.135. Multiplying this by our initial 1,000 gives a final answer of 135 molecules. Because 135 is greater than the threshold, this cell will successfully transform into a motor neuron. Having calculated the chemical concentrations, let us visualize how these processes come together. On the left of this diagram, you can see the neural tube, with its top end expanded into the 3 primary brain vesicles: the forebrain, midbrain, and hindbrain. Next to it, the red shaded box represents the tissue of the neural tube. with the dark red color at the bottom indicating a high concentration of a morphogen, like Sonic Hedgehog. As you move upward, the color fades, representing the decreasing chemical signal. The graph on the far right plots this exact relationship. You can clearly see the exponential decay curve we just calculated, showing how the concentration drops off as the distance from the source increases, ultimately guiding the fate of every single cell. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that the entire nervous system begins as a simple flat sheet of cells that folds and fuses into a hollow neural tube. Second, the top of this tube expands into primary vesicles, laying out the physical architecture of the brain. Third, we saw how stem cells use rapid exponential growth to generate the billions of neurons required for a functioning brain. Finally, we discovered that precise mathematical gradients of chemicals, like Sonic Hedgehog, act as a map, telling each newly born cell exactly what type of neuron it needs to become. You now understand the foundational steps of how the most complex organ in the universe builds itself from scratch.